University of Virginia. Professor Blemker did her PhD at Stanford University with Professor Scott Delp, and she has a research program that's primarily focused in the mechanics of skeletal muscle across physical scales. And I was very happy to have her accept this invitation because I think her research is very close in terms of the general modeling paradigm to the types of things that we're interested in this course. In particular, she's very focused on modeling and simulation across multiple scales, along with complementary experiments. And with that, I'm going to turn the podium over to Professor Blemker. And thank you again for coming, Silvia. I really appreciate it. Yes.
That's good. Hello. Well, it's exciting to be here. I haven't been to this conference before, or this workshop, though I've had a few students come in the past, and Adrian Williams here is here representing the lab this year. So I actually went, I came, do you remember this, Jeff? I came out to visit you when I was a graduate student. Do you remember this? When I was starting this talk, I went through, you have your long list of presentations, and I have this Utah 2003 talk. So I was just curious and opened it up. It was pretty funny. But I'm interested in computational modeling of skeletal muscle, and I got excited about the potential for finite elements and continuum mechanics as it applies to skeletal muscle. At that time, people weren't really using continuum-based approaches to modeling muscle, and actually even at that time, Jeff was really valuable to me as I developed some of the constitutive framework that we're still using in the lab. So before I dive into the talk, I also wanted to give a little plug for University of Virginia and the Biomedical Engineering Department. As Jeff said, I'm a primary in the Biomedical Engineering Department. I have courtesy appointments in other departments, mechanical engineering, orthopedic surgery, and ophthalmology. And so that's a reflection of the interdisciplinary nature of our program and the fact that our BME department sits both in engineering and medicine. So it's a really exciting opportunity for students to train in an environment where they get to interface with lots of engineers, but also be in clinics or even work with basic sciences in the School of Medicine. So we're always recruiting graduate students, postdocs, faculty, so keep us in mind as you move on in your careers. So back to my title. I have this sort of long title that is a mouthful, but I decided to retitle it a little bit kind of when I was sitting in the room this morning, as you guys were going through a lot of mechanics this morning, a lot of equations. And so if, you might have been thinking to yourself, what did I get myself into? Why am I doing this? So I decided to retitle my talk, Why Model? I'm going to talk a lot about the different types of modeling projects we've done in the lab and some of our techniques, but there's going to be a flare of why are we doing this in the first place, because I think it's always important to keep that in mind, both to motivate you for doing all this stuff, but also to really focus your modeling efforts to gain the key insights that you want to gain. So I'll start with just that question, to model or not to model. So why even model in the first place? There's, in the world of data explosion, where we could be out there collecting data, why are we generating our own data instead of going out and collecting data? Well, I'm going to first quote Hamlet. So Hamlet asked to be or not to be, right? So he was thinking in the context of life having so much hardships, why even live at all, right? He was very pessimistic view on the world, and so you could think the same thing in model. Is it worthwhile to create models when they contain so many hardships? I saw all your. I love sitting in the back there and seeing. I saw a bunch of screens of like FE-bio numbers coming through, and I just, you're sitting there crossing your fingers. Is it going to work? Is it going to converge? Blah blah blah. It's the highs and lows. So why even do it? Is it worth it? Well, Hamlet would say due to fear of death. So maybe our analogous statement would be due to fear of experiments. I don't know. Some of us don't really like to use our hands very much, and we want to stick with the computer. So that could be a real thing, but maybe that's not the best answer. So maybe an alternative answer to maybe Hamlet's, depending on what your faith in life or whatever is to live out our purpose in the world, have an impact. So we're here because we want to have an impact. Similarly, maybe we're using our models to maximize the impact of our work. So that may seem obvious, but I think it's important to realize all the time is that really the point of the modeling is to maximize the impact and relevance of the science that we're doing. And if you're thinking about where your work is headed. So in the context of the modeling that we do, there's a few ways in which modeling can maximize the impact of research. So one way I think about it is the scale of these guys are sort of are kind of reminiscent of how I sort of think about models. You know, if you think about what we know about for example, the human body, it's still pretty small, right? There's a lot that we don't know about biology and biochemistry, all these things that govern how our body works. But in a model, we can sort of pretend like we know a lot. We stick everything we know in the computer, and now we know a lot, but we can also really dive into it and study it in detail. And we can do things like tease things apart to explain an experimental finding. We can put in enough in the model that can replicate an experiment that maybe can really understand what's going on. We can uncover cause and effect relationships, do what-if scenarios, simulate treatments or surgeries or whatever it is. Also estimate quantities that are not measurable. So oftentimes there's just only so much you can measure, whether it is that you're studying humans and there's only so much you can measure in a human, or maybe it's just something you're interested in, something that is simply not measurable. A model can give you insight into that. Or determine congruency between experiments. Again, now that there's so much more experimental data out there, also so much more issues with reproducibility, where one paper says one thing, another paper says another, models can be used to really figure out who's right and who's wrong, or maybe both people are right and they just did it differently or whatever. But models can be that linker between experimental studies. But ultimately, what we're trying to do is kind of do all this really kind of cool, interesting stuff, but lead to things that actually drive a field forward, to lead to unforeseen hypotheses that ultimately could inform new experiments or treatments. So kind of thinking forward and saying, okay, the ultimate goal of the modeling is actually to move out of the modeling realm and do something that's going to affect the outside world. And I would say that these are the success stories. So as much as we can strive to do that in our research, but also when we see it otherwise, really think about those and use success stories of how a modeling project, a modeling study led to a new treatment or solved a specific problem. Looking for those in your field or outside fields, and realize that those are really important in order for us to advocate the importance of computational modeling in medicine. So I also, because I found that 2003 talk, Jeff, I was like, okay, well maybe I'll talk a little bit about where I came from and how that led to the research that I've been doing in the lab. So what led me to go into finite element modeling? So as Jeff said, I came from a lab of Scott Delp where nobody did finite element modeling in that lab. It was all kind of rigid body simulations of movement and simplified representations of muscle. But there was a student at Stanford who did some interesting experiments in muscle with a new dynamic imaging technique where he was able to measure the motion. So this is an arm, and this is the humerus, and then this tissue over here is the biceps muscle, so the muscle that flexes your arm. And so he was able to measure the motion of the tissue as a person inside the MRI scanner moved and flexed and extended their arm. And so what he could do then is look at how the muscle shortens in vivo. Before that, nobody had been able to make such a measurement. And so before that, all the models that had been developed in muscle assumed that muscle shortened uniformly. It was kind of one rubber band that all shortens the same. But if you look at these regions, so, sorry, I'm using this laser pointer. I hope it's not distracting. I only have one side. But if you look at how these regions, so these are tracking regions in the tissue, these overlap a lot, these don't as much. So it's showing that there's a lot more shortening in some areas of the muscle than the other. So that led to my work of saying, well, okay, we want to explain that finding. We can't do that with the simplified models of muscle that assume everything's the same and that it all shortens uniformly. And that led us to move to continuum representations of muscle. So that was developing a finite finite element model of the biceps muscle is a very simplified geometry at that point, and predicting, so change in length or strain as a function of length along the muscle. The shaded is the experimental data, and the dashed line is the model. I was able to develop a model that replicated that experiment and then go into that model and figure out what about the specific architectural features of the muscle were contributing to that strain deformation pattern. So that's where I started. So that was like that first bullet of explaining an experimental finding. I guess then I just got hooked and stayed and doing this kind of thing. One thing that got me hooked is that muscle is really hard to model correctly. It's really complicated. So it has lots of interesting features. So I'm showing a microscope image of a muscle cross-section. So these are muscle fibers. Around them is extracellular matrix. There's lots of other cells in there too. The ECM is made of collagen. Inside the fibers, the contractile proteins. So there's so many things about the tissue that make it really complicated from a continuum mechanics standpoint. Of course, it has, it's nonlinear, it's nearly incompressible, it's biphasic, it's got active and passive elements, it's non-isotropic. You might assume it's transversely isotropic. You might question that. Who knows? But it's definitely not isotropic. There's neural innervation. There's rate dependence. There's lots of things going on. So to some degree, it's an interesting field of study from a continuum mechanics standpoint because it presents so many interesting challenges. So a little bit about the constitutive model that I developed a long time ago, and that the lab we've been using variants of that ever since, been developing. It's a transversely isotropic model. It was kind of fun that you already were talking about some of this morning, so I don't have to talk too much about it. But using hyperelasticity theory, so we define the stress tensor as a derivative of the strain energy density function with respect to the right Cauchy-Green deformation tensor. And we use an additive form for the strain energy. So one somewhat unique thing about this model is that the invariants it uses are slightly different from the invariants that Dr. Weiss talked about this morning. He talked about I1 through I5. We use a slight variant of that. So one of them is similar to, is the lambda stretch, the I4, or square root of I4. And so that's representing the behavior of the muscle in tension. Along the fibers, and so that's where the contraction happens. It's a muscle shortens along the fiber and activates. So we can then incorporate the known properties of muscle fibers in that direction, where we have a passive and an active term that are both dependent on stretch, and then the active term is going to be dependent on an activation state, which is an input to the model, which can be time varying throughout a simulation. But then we also defined two other invariants that were originally introduced by Krishon in 2002, so right around when I was working on this model, where he defined, instead of I2 through and I3, instead of I2 and I5, he defined a term that represents a long fiber simple shear. So if you wanted to represent the deformation simply along the shearing along the fibers, it looks like this, and then a term that's defined as a cross fiber shear, so deformation or shearing across the fibers. And what motivated me to do that is the fact that I wanted to really be able to isolate the properties of muscle in those directions. For example, shear properties was of interest because muscle fibers are connected by this connective tissue stuff in between the fibers, and that resists shearing along the fibers. And there have been a lot of debate in the muscle literature about what is the role of that shearing in muscle. So I wanted to be able to have a knob to turn to study how shearing affected, but with the kind of the standard invariants, there isn't a specific term that represents that. So that's the elements. I'm not going to, so we've been iterating on this model for a little while. Originally, the relationships here were nonlinear. Are linear, now they're nonlinear, and we have a bunch of other things that we've changed about the model. But that's the basic form. The other thing that I think is of interest to the group is kind of the challenging problem of how to model what we call muscle architecture. So if you look at different muscles across the body, they have different arrangements of the fibers. So for example, this tibialis anterior muscle has a kind of simplest version where the fibers go from the top to the bottom. They're relatively parallel with each other. But we also have more complicated architectures, some where the fibers actually insert into a long tendon, so there's sort of a variation in the length. There's also bipinnate muscles that have angles. They go from like one side to the other, but then they kind of insert into a middle tendon, and even multipinnate muscles. So there's lots of different arrangements of fibers within a given volume. So you can imagine if you're creating a model from image data from an MRI, you could get the representation of the 3D surface, but we also, in order to utilize this constitutive model, which is transversely isotropic, we need to be able to model how the fibers are mapped within that muscle, and every muscle looks different. So, so for in the heart, you know, essentially people know where the fibers are and how they run, so you can kind of represent that. Not easily, but kind of in a, it's pretty streamlined in order to represent that. But whenever we create a model of a new muscle, which we're doing all the time, we have to figure out how to represent its specific architecture. So this is actually something that Adrienne is working on. She put together this nice slide that illustrates the different approaches that one could use, which are also kind of relevant in the cardiac modeling and other areas in which modeling tissues that have fiber structure. So there's, if you think about, we sort of bend approaches into three: data-based, atlas-based, and rules-based. So, and they're not totally mutually exclusive either, so that's why Adrienne drew this as a kind of a Venn diagram. So data-based might be making use of DTI measurements of fiber architecture and inputting that into the model. Though, if anybody's worked with DTI data, you know that it's relatively noisy, especially in muscle. You don't have nearly the signal-to-noise ratio in muscle as you do in the brain. So it's quite noisy data. So you have to process that significantly if you want to get to a fiber field that's smooth enough to run a simulation. Or the groups have done some really painful dissections of muscle fibers, dissecting fibers and digitizing them manually all throughout a muscle and getting a 3D cloud of points, which sounds pretty painful, and really you can only do that on a cadaver or an animal. The next one is a atlas-based approach, which is what I used previously and what many others have, is you define kind of a template mesh and fiber geometry, and then you map that to a specific mesh. So you're sort of, you have an atlas of different types of muscle fiber shapes, and then you can map that on. But then the third one is this rules-based, and we're the method that we've started to adopt in the lab, we would call rules-based, which is more saying things like, well, we know muscle fibers originate on one end and terminate on the other, and we know that they behave in certain ways, so let's model the muscle that way. And it turns out that you can use fluid simulations to do this, which sounds pretty strange. So we're modeling muscle fibers with fluid dynamics. That makes no sense whatsoever, but maybe after this slide you'll understand. So the way that we're thinking about it is that the properties of a viscous laminar flow resemble those of the way muscle fibers look. So for a laminar flow, it's consistent because fascicles will not reverse directions abruptly. Viscous, because fascicles are coaxially aligned. For example, you know, this would be, for the laminar example, this would be a turbulent flow versus laminar, and we know fibers look more like the laminar case than the turbulent case. And incompressible, fascicles don't branch. So fascicles, as they're moving, they stay next to each other. You don't have like holes inside the muscle. So those are, that's sort of how we've start to think about why a fluid simulation might resemble that of a muscle. Actually, and I've spent a little time talking to developmental biologists about how muscles form in utero, because they actually do form, fibers extend from the tendons and merge. So there's some sort of notion that they could sort of flow towards each other, but that's a very hand wave situation. But here's what it might look like. So here's the hamstrings muscle, the biceps femoris long head, and the leg. And here's an example of how the fibers flow. So it's sort of illustrating the path of the streamlines in the field. So they originate on one side and they're flowing and insert on the other side. So this is an example that Adrian has developed of the gastroc model, a muscle, gastrocnemius muscle in the calf, where she has a 3D model that she created from MRI data, and she's defining the inlet and outlet conditions of her flow field based on where the fibers attach. So we know the fibers attach into two tendons, and actually sometimes we call those tendons aponeuroses because they're not just an external free tendon like you might imagine the Achilles tendon. They're actually tendons that come into the muscle and the fibers insert into them. So they're kind of tendons within the muscle, but the fibers insert there. So there's the inlet where the fibers start and then the outlet where they go. And then here's what a flow field might look like, where you can see the inlet condition here and the outlet condition, and the color coding here is actually fluid velocity, which we're not sure actually means anything or not. We're sort of curious whether it does, but what we're most interested in is the streamline that comes out of this analysis. And then Adrian's been validating her models by comparing it with whatever data she can get her hands on. For example, this is comparing the distribution of fascicle lengths that she gets from her model. So it's a histogram of fiber length. So you can imagine with a complex shape like this, you're going to have some really short fibers, some really long fibers, and some in the middle. And she looked at her means and compares it to literature values, and they're right on. They're looking really good. She's also in the process of looking at ultrasound as another way to validate what she's getting from her model. So because it's a rule-based approach, we need to cross-validate it with other experimental measurements. Otherwise, it's just kind of a made-up thing. But most of our comparisons have looked really solid. There was also another paper that came out of a former PhD student in the lab who's now in New Zealand, where he compared what he gets from CFD models with DTI data, and he gets very favorable comparisons. Okay, so that was a little bit about the modeling, kind of the technical aspect of, somewhat technical aspect of the modeling that we do with the constitutive model, how we generate muscle models, and how we look at fiber architecture. So that's sort of, what the neat thing is that you can apply that to lots of different models, or muscles. So I wanted to bring up another idea about thinking about a path of a modeling project or modeling work, or even your career, I don't know, just where you go with research. As you know, research is sort of unpredictable, right? One thing leads to the other. And so how do you think about the impact that you want to have with the modeling work in the context of the broader picture? So I've kind of made a plot that shows the relationship, or maybe lack thereof, I don't know, of what your desired modeling scientific impact might be, or your desired non-modeling scientific impact. So modeling would be like a development that you've made that other modelers will be really excited about, like how you solved you have a new element that really solves the incompressibility problem. Like you don't get all the issues that you normally get, like, ooh, that's super exciting. But other people wouldn't even know what you're talking about, right? So that's like the new modeling ideas would come here, and then there's the non-modeling, the things like having an impact on a clinical problem or a basic scientific question. So something that's your modeling output is of interest to a community that's not modelers. Interestingly, because I did my PhD in a modeling lab, I actually didn't really realize that these might be two different things. I never really had to justify why model ever. Nobody really questioned me until I became a professor, and I had to write grants and papers, and people questioned it all the time. So then I realized that there's a bigger world out there that you need to know about and understand. So okay, so you can have, as we do as a modeler, we do both of these things, and you can imagine that as you're deciding on a project, you might sort of choose a path, different types of paths that that follow this landscape. For example, you might have that the first step that the work that I just described of developing this modeling framework, I would say was primarily a modeling effort. I gained some kind of interesting insights about that muscle, but really a lot of it was like developing a new modeling framework. So I would call that modeling scientific impact. But then it's like once you have that, maybe there's some other really cool things you can do with it that can maybe not necessarily advance the model themselves, but maybe apply it in new ways that can have an impact. And so you might imagine continuing with that, and maybe you get to a point here where to get to the next level of clinical impact, you need to make a huge modeling development. So maybe you stop, or maybe you go sideways, develop, make a new modeling method, and then ultimately accelerate out. But I think what we all want to do is go out this way, right? And one could argue that these should be totally correlated with each other or not. I don't know. Even are these independent of each other? I don't know. We could talk about that philosophically a lot if we wanted to, but I just wanted to bring up this concept as you're deciding on a modeling project where you might be going in this landscape. So a couple of examples of how in our lab we went from that horizontal line, which is purely new models, to more of a vertical line. What's neat about muscle modeling is that there's muscles everywhere. We have muscles in our eye, we have muscles in our body to make us breathe, we have muscles in our arm, have muscles in our mouth to make us speak, and our legs. We have lots of different things. There's actually lots of really exciting applications of muscle modeling work. So I thought I'd give a couple of examples of ones that maybe are not as commonly thought of. Usually when people think of muscles, you think of leg muscles or arm muscles. So I wanted to bring up a couple of examples. So one of them is muscles in speech. And specifically the clinical problem of cleft palate repair surgery. So it turns out in the roof of your mouth, there's muscles. So there's bones and muscles. And a cleft palate can be combined, is essentially a hole in that area. And it can be combined with a cleft lip or not. But the cleft palate is really the hole that's in the roof of the mouth. A child is born with that. It's a congenital birth defect that needs to be repaired. So if you don't have a hole, if you have a hole in the roof of your mouth, it creates a fair amount of difficulty. Number one, you can't really eat. And two, you can't talk, really. Because if you look under there, there's the hard part, but then there's some soft stuff, which is called the soft palate, where there's some important muscles in there and soft tissues that are required for eating and speaking. So in particular, there's this one muscle called the levator veli palatini, which is back there, and it's a sling-type muscle that moves the soft palate up and down as you're talking and eating. So if you look, this is now like a sagittal section of the head. So here's the mouth, here's the tongue, which is enormous, by the way. It's a crazy muscle. So here's the soft palate. So there's muscles going in here, in and out of the page here, that move that soft palate up and down as you talk. So as it's contracting it, so it's coming out of the page, and it moves this thing up and pushes it against the back of the throat. So it turns out for most of the sounds that we make as we talk, we need to pull this thing up and form closure. It's called closure between the soft palate and the back of the throat. So it's closing off the airway between the nose and the mouth, because most of the consonants and sounds we make require oral pressure. There's only a few M and N, really, that don't require that kind of closure, and those are called nasal consonants. So, of course, if you're born with a hole there, it's hard to make closure when there's that big hole, but also that muscle is completely misaligned in a child that has a cleft. So instead of forming a sling like it should, it actually inserts directly into the hard palate, which is also has a cleft. So the surgery needs to rearrange that muscle, needs to separate it from the hard palate and then stitch it back together so that muscle can move. It turns out that that'll be done on a kid, so that a child's born with it. They'll do this surgery at about 10 months of age, but so usually the feeding is pretty good after that, because they've covered the whole hole. But come when the child becomes like 3 or 4 years old, you realize that many of the kids end up with a speech problem. So that muscle actually doesn't reform enough to really be able to move that soft palate enough to form closure during speech. So in normal, it's called velopharyngeal function. That's when you use that muscle, you can form closure with the back of the mouth. But if you can't form closure, then it'll maybe move a little bit, but it won't form that. And then you end up with a pretty significant speech problem where the children end up being quite intelligible. There's a whole metric system for speech therapists to assess that, but it's still a pretty big problem. So we aimed to say, okay, well, this is a muscle. It deforms, it's stretching tissue. We can model that. It's the same constitutive model. It's a new geometry, but we can do that. So we looked at one particular research question I'm going to talk about here. We've done a lot of different research questions with this project where we said, well, okay, well, here they're reforming the sling. Well, how do they know how much to tension that muscle to give the proper function? How does the tensioning of that muscle influence its ability to form closure? So we did a number of simulations where we created a model, but then we simulated different amounts of overlap or tensioning. And we ended up, so here's some examples. This is sort of a simplified version, but we've been collecting a bunch of MRI data of both healthy and children with cleft, created 3D models now, actually including some of the other muscles that are involved in the motion of the soft palate, though this levator veli palatini is the primary one. And here's one example of the outcome where we said, okay, if you simulate different tensioning, there's actually a sweet spot of overlap that gives you the maximal force-generating capacity of the soft palate to form closure. And interestingly, most of the surgeries, if you were to, the techniques of dissection and repositioning would have put the muscles really at no overlap or maybe even separation. So that we thought that was pretty exciting. It's a modeling prediction, a cause and effect, or sort of simulated surgery. So we got, it's pretty interesting, we thought. So I also wanted to give you, I have a few points in my talk where I give you like anecdotes about what it's like to like publish, because a lot of times when people talk about their research in a keynote or whatever, it's like seemed like everything was so perfect, right? Like, oh yeah, this all worked great. I knew what was happening and whatever. And that's not really the case. So, all right. So we had this preliminary model. I meant to say that the work, because I had no pilot data, I could have never gotten NIH funding to do this project. But I did find a foundation that was willing to take a chance on us. I could tell a compelling enough story that we had the right expertise. And they gave us some money to get the project started, so that was exciting. So we got it started, so we tried. Our surgeon collaborator gave us the go-ahead to submit to the top plastic surgery journal, and so we got these reviews that were, they're pretty typical, but your assumptions and correlations were not validated. The results are based on the results, and conclusions are not warranted. All about the assumptions, the viscoelastic properties were incorporated. So I kind of restated these. Your model is not validated, your model is too simple, and your output doesn't make sense to me. So it was a reject. Oh well, back to the drawing board. So we stopped. No, we didn't stop, of course. You got to move on, press on. All right, so it was rejected. But then, we continued to refine the model, and we hadn't even presented the work yet, so it was sort of our first sort of soiree into the field. We presented the work at the annual meeting, the Cleft Palate Craniofacial Association meeting, and we got a lot of really exciting feedback. We refined the model, then we published it in a different journal. That was exciting. And then actually, just recently, a paper just came out that proposes to do a higher amount of overlap than anybody had ever done with really a high success rate. And I'd like to say that they did that study because of us. I don't think that's the case. It happened, so this guy, Albert Wu, is a surgeon who was at WashU, now he's at Brown. He had sort of figured out a way to dissect the muscle where you could achieve more overlap than other people had. And so we consulted with him a fair amount as he was developing the procedure and trying it on kids. It's amazing that surgeons can just like try stuff you know? But he did it, and it looks like it's working really well. And so, and now we have funding from the NIH, a project that, you know, from the very beginning, I didn't have anything to do. I had nothing. I barely even knew like that a cleft palate was different from a cleft lip. And now we have NIH funding for it. So it sort of speaks to you can reinvent yourself, try new things. You have to be a little bit crafty about how you get things going. You have to be sort of relentless, but you know, it can work out. Just another anecdote of a project. This is a project that this time it was an area that we'd never done before, and I hadn't even thought about, but somebody approached us to work on it, a startup company called Ace Vision. They have a treatment for presbyopia, which is the need for these. So like whenever you guys turn 40, you're going to need these. Everybody gets presbyopia, the inability to change your focal point. So as you age, it's just harder and harder to change your focal point to near. So it turns out it's a muscle that does that. It's called the ciliary muscle of the eye. And this company has a new sort of treatment. It's not a surgery, it's a treatment that aims to address presbyopia, and so you wouldn't, without the need for eyeglasses. So you can imagine the market's quite big if you think about that. But they're kind of, their viewpoint is very different because normally in ophthalmology, people don't think about muscles being important for presbyopia for whatever reason, even though it's a muscle that's involved. So they came to us to develop a model to kind of explain and explore the mechanism of their treatment. So we developed a 3D model that got quite complicated. This is the muscle in here that has 3 fiber families. Here's the lens of the eye, here's the cornea, here's the scleras outside of the eye. The muscle is attached to this lens via a network of what are called zonules that are kind of like really tiny little tendons. We can simulate ciliary muscle contraction. We compared with a bunch of experimental data where the deformation of the muscle is measured with this really small ultrasound probe. And we got results. So I'm speeding along, but this is another interesting setting where, because it's funded by a company, ultimately they just said, well, how should we design our clinical trials? And you can imagine my graduate students kind of like nerves, like, okay, wait, I'm supposed to tell them what to do based on my model? So it's been an interesting, so she actually helped design a preclinical, which is a monkey experiment that they're doing. And you know, the point that we realized is that it's actually much more informed, the clinical trial is much better informed with a model than it would have been otherwise, because she needed to figure out, essentially her treatment is a set of laser ablations to the external part of the eye, the sclera, to make the sclera tissue more compliant, which allows the muscle to contract inward. But she had all these potential patterns to do and all these potential densities of treatments. And what Katie was able to do with her model is essentially narrow it down and show her, okay, you don't need to try all these. You only have 6 monkeys. You can't try like 8 different things. But here's what a difference you'd expect to see. And she even powered the study off of her modeling prediction. So it's another kind of interesting example of how you can push your model to do something relevant, maybe a little bit uneasy, but at least it's a preclinical trial before the clinical trial happens. Just the last tidbit I wanted to show, kind of going back to this, is these are kind of nice examples of where we went more, where we kind of veering up to the clinical impact. And so how in a research program you might be sort of straddling all of these when your fundamental is kind of expertise is in the modeling. So I thought I'd give a little overview of another project, a relatively big project in the lab on Duchenne muscular dystrophy, which this is really one of the projects that does this multi-scale, as Dr. Weiss was mentioning, where we have to consider multiple scales to really tackle the problem. So Duchenne muscular dystrophy is it's a disease of the muscle, another genetic disease, which is caused by a mutation in the gene that encodes for the dystrophin protein, which is a protein that connects the contractile apparatus of the muscle. So this is a sarcomere. So this is where the muscle force is generated within the muscle fiber. It connects that all the way out to the extracellular matrix. So it's a linker protein, essentially. And so boys that are born without this dystrophin protein have what's called Duchenne muscular dystrophy, which is pretty bad. So the boy, you might be first detected when they're about 3 to 5. Otherwise, they seem somewhat healthy, but they're having some difficulty with walking and standing up. But then soon after they're diagnosed, they become weaker and weaker and have dramatic changes in their walking pattern. If you look inside the muscle, so that would be kind of a macro scale observation. If you look in the muscle, you're starting to see the muscle doesn't look as healthy anymore at the tissue level. The fibers are getting smaller. You have connective tissue in there. You have a bunch of inflammatory cells. And then ultimately, by early teens, the boys, it only affects boys, will be in a wheelchair. And if you look inside the muscle, it looks very damaged and diseased. You'll have fatty infiltration, connective tissue, necrotic tissue. And boys with DMD they now can live into their mid to late 20s, but usually wouldn't live past early 30s because it affects all the skeletal muscles and even the heart. So it's a pretty devastating disease. Interestingly, it's kind of initial source of the initial issue is just lacking that dystrophin protein, which is primarily has a bunch of functions, but primarily is thought to mechanically protect the fibers from damage during contraction. That's sort of thought as its main role. And so what people think is that because the muscles are being used, that they'll get damaged over time much more than a healthy muscle would, and that's what leads to the degeneration. So one question that we thought about early on, so I've been interested in this problem for a long time, and I'll tell you the kind of anecdote of how the project developed in a minute, but one of the questions we got compelled by that we thought models could be useful for was why do some muscles degenerate more quickly than others in DMD? So actually, if you look at an MRI scan, so of a healthy person, it's a healthy person, all the health muscle is dark. That means in this type of scan, that means that it's healthy. But this is the MRI scan. This is a thigh of a boy with Duchenne muscular dystrophy. So the white pixels indicate fatty infiltration, which is thought to be the marker of damage. So as the muscle becomes damaged, it'll essentially get replaced with fat tissue. So interestingly, if you look across this one cross section, you see some muscles that are highly degenerated, highly replaced with the white pixels. Some that almost have no white pixels whatsoever. They look pretty good. And then some muscles in the middle. So all of these muscles lack the protein the same way. They're not different from each other when you look in a microscope, but the manifestation of that disease actually varies between the muscles, and even though they're kind of next to each other. Embryologically, they're similar. Lots of things are similar. So, we're developing models to kind of tackle that question because we feel like answering that question could have lots of important implications on treatments for DMD. Right now, there's literally no treatment. There was the first FDA-approved treatment, but it doesn't actually have any clinical benefit. So so yeah, there's a whole story there. So one of the areas that could really impact this would be movement biomechanics. So I included this. I know that there's a few here that are interested in open sim and musculoskeletal simulation. Is that right? I remember talking to a few people. So. So that's sort of a macro scale, but then there's also some microscale differences between muscles that could lead to this pattern. But we know that dystrophic muscles, as I said before, are highly sensitive to what's called eccentric contraction-induced damage. So when you use your muscle eccentrically, that's when you're using it when it's breaking your limb or decelerating. So you're activating the muscle, but it's also stretching. So that's sort of like if you lift up a weight, and then as you let it go, it's harder to let it go softly, and that's because you're using that muscle to slow it down. That's called an eccentric contraction. So it turns out that even in a healthy, so the control, these are experiments done on mice. Eccentric ECC would be eccentric contraction protocol. Even in a control, you get a little bit of damage, and actually that's good for a healthy muscle, promotes remodeling. But in the MDX, which is the mouse model of muscular dystrophy, you get a lot of damage from that, and much more than the other types of contraction, either isometric, you just hold it fixed, or a passive stretch, or just over time. So the eccentric contraction is bad. We also know that in walking, we use some muscles eccentrically more than others. Some muscles, when you're moving, have to break the limb, decelerate the limb during the swing or stance phase of walking much more than others. And this is a metric of that. Negative work would be the work is defined as the force times displacement. So negative work would mean that the force and the displacement are opposite, which would be eccentric contraction. So some muscles have a lot of negative work and some don't. So we thought to correlate these to see. So we used this open sim model of walking that was actually already published. So that made it quite nice. Another kind of nod to the utility of open source and sharing models is that we were able to use a model that was already established. And based on it, we could quantify the amount of negative work during gait of all the muscles of the leg. And then we could compare that with the percentage of fatty infiltration that had been observed in a number of studies. So then we aimed to correlate them. So the question is, did the fatty infiltration relate to the negative work? And lo and behold, there's a correlation there. And that's even using data from average data from one data set compared to walking motion of a healthy individual. So that shows that there's potentially something there. So in general, the muscles that are damaged more are also the ones that we use eccentrically during walking, with the exception of, here's one outlier, and that was for the lower limb. The same thing holds for the upper limb as well. So that gave us some interesting results. Essentially, great pilot or preliminary data for a grant, essentially, was this. But the other thing that it's good to do the negative control. So if you compare the fatty infiltration with the positive work during walking, which would be the muscles used kind of the way you more typically think about them shortening, then you don't see a correlation. It's kind of a nice negative control. The thing is that there were those outliers. And now what we're interested in is the fact that actually, if you look within the muscle, this image sort of shows it, but there's some other nice data that shows that actually you look at the distribution of fatty infiltration in the muscle, it's highly inhomogeneous. Adrian is working on developing models, finite element models, and relating that to the pattern of degeneration to see if within a muscle that the regions of more degeneration also correlate with regions that undergo more stretch in the muscle. So a couple of other things that we're doing, kind of speaking to the multi-scale, is that was sort of a macroscale thing. We've also gotten interested in the microscale mechanics of DMD muscle. So if you look at images of muscle through a microscope, and a healthy muscle looks nice and filled with muscle fibers, but in the dystrophic muscle, of course, you see lots of variability in fiber size, and you see this fatty infiltration. So we are interested in modeling that and understanding how the microstructure influences the tissue mechanics and the damage susceptibility. So we developed models of muscle cross sections. I'm just going to where we wanted to model variability in the cross section of the muscle. So we modeled things like variation in fiber CSA, fibrosis, and fatty infiltration. But we also actually included some effective anatomical variability, sort of stochasticity in different shapes of fibers, so that to make sure that our conclusions weren't just sensitive to like the specific geometry we were looking at. We delved in a little bit to the micromechanics. So you're just sort of in FE Bio, you can do some multi-scale analysis right now. You have like a micro model that links to a macro model, right? So this is sort of focusing on just the micro model for now, is looking at how the properties of this is a fascicle or bundle of fibers influence the tissue properties. And we're actually including links that represent the function of that dystrophin protein. Remember I said it connects the fibers to the ECM. And so specifically we're looking at it in shear. Here's the model in shear. We have a boundary layer that allows us to kind of study lots of different shapes of muscle fascicles. and not worry too much about periodic boundary conditions. So we define the properties of the boundary based on a mixture theory. And, sorry, I'm just zooming through here. I want to get to the end. So we can also calculate the membrane strain. So that is our kind of surrogate marker for damage. And Dr. Weiss was mentioning before that one of the problems or kind of challenges in biomechanics is modeling damage, right? Now that we've figured out how to model kind of normal or sort of undamaged tissue, now to the point of being able to model where and how is tissue or cells damaged. So this is a relatively simple approach, but knowing that here we're primarily interested in damage as a result of the membrane, because we know that's membrane stretch, because we know that's mechanism of the disease. So we can measure the shearing of the membrane and look at how things affect that. So this is one example. So in a healthy muscle, during a shear deformation that would be consistent with the eccentric contraction, we get a little bit of membrane strain, which is reasonable. But if we remove the transmembrane proteins consistent with the lack of dystrophin, there's still other proteins that exist there, but if we remove some of them, we get an increase in membrane strain that is consistent with the fact that the muscle is damaged easily. But then if you add the fibrosis and other effects, you get a little bit more membrane strain. Actually, the inclusion of the fat infiltration actually kind of normalizes things out a little bit here. So late, and well, of course the muscles aren't being used late anyways, but they wouldn't be as susceptible to damage as the kind of early phase. One interesting thing, though, is that if you modify the compliance of the ECM, so this is assuming a relatively stiff ECM, but if it's more compliant, you get a great increase in that damage susceptibility. So back to our original question about why some muscles are not others, it turns out that the ECM properties vary substantially across muscles. Some muscles have really stiff ECM, some not as stiff. For example, it turns out the extraocular muscles are very well preserved in boys with DMD, but and they actually have a very unique extracellular matrix structure and link with the fiber. So this is somewhat consistent with that. Okay. I don't usually do this. For some reason, this is longer than I thought it would be. I wanted to bring up one other thing about predict. So that was kind of the microscale mechanics, looking at how differences in the microscale structure might influence damage susceptibility. So we had a macro and a micro. But there's also cellular factors, of course, at play. The dystrophin protein has a lot of functions. So we're creating a modeling framework that allows us to not only predict the damage, but predict what happens afterwards. Because there's a lot of mechanisms at play, like the contraction-induced injury susceptibility, but very pro-fibrotic states of cells that are trying to create fibrosis, chronic inflammation. The satellite stem cells, or the stem cells of the muscle, they have ulcer dynamics. So we're linking micromechanical models with agent-based models that are capable of predicting the cellular responses as a result of the damage. There's a few people in here that are doing agent-based. Who's doing agent? Is it just one person? All right, I'll do a very quick introduction of what that is for those who are curious. So in this context, our agent-based models simulate behaviors and interaction between cells, which are, quote, agents. So agents have autonomy, right? Or agency over their behaviors. They have rules that govern what they do, kind of not too much worried about everybody else, just they affect each other through their interactions. So, for example, a fibroblast that might exist inside a muscle, that's a specific cell that produces collagen. It can be sensitive to things like different growth factors that are in the environment that are diffusing neighboring it. If it senses them, it might decide to proliferate or migrate towards a factor or differentiate into something else. Like a fibroblast might turn into a myofibroblast, or secrete other factors, or die. And so in an agent-based model, we can incorporate rules, variety of rules for each agent that are based on experimental observations, like fibroblasts in the presence of a specific cytokine decide to differentiate. and we can model those things, and they have some stochasticity associated with them. So there's sort of rules. It's a rules-based model that then we can put them together and get observe an emergent behavior from the multiple agents that are within in a model. So our model looks like this, where it has the fibers and it has the ECM. We can base it off of histology, and we'll have a lot of different things like different cell macrophages, inflammatory cells, the satellite cells, and here's just a little illustration. So if we model the damage, we can model how the muscle regenerates following the damage. So it's a time-varying phenomenon, and we can simulate the dynamics of cells within that model and predict how they're changing over time. And so, of course, model validation is always an important piece. So we do different things like surveying the literature or doing our own experiments. We can track things like cell counts. So, for example, we're here interested in fibroblasts because they promote fibrosis in the muscle. And so we're plotting how that changes over time after an injury. And then we can compare it to the model plot. One cool thing about these models that's different from an FEA model is that there's a shaded region. So this is the model, and that's because the rules are stochastic. You're not always going to get the same answer from the simulation. So that's very empowering when you're doing analyses. And then we can do different things like cause and effect. For example, see what happens if you are in a pro-fibrotic versus a not pro-fibrotic state. And then, or test potential therapies, like a membrane therapy that exists in the field or potential in the field right now, and how that would predict the ability for the muscle to return to recover. So just to finish, sorry, I'm a little long today. We're using computer models to illuminate disease. So in this particular application area, it's exciting because we're kind of looking at all these different scales, and we've had some nice insights into effects of the cellular microenvironment, biomechanics, and even movement biomechanics and mechanical loading. And so we're ultimately now have a 5-year grant to link these scales in a modeling framework and have kind of a multi-scale analysis that's able to account for these interacting mechanisms between the macroscale and the microscale. So this is a project that I wanted to get going on when I first started as a professor. I wrote my first grant on it when I think I was my second year. And I think I got triaged first. Then I tried again. I think I got like a 50 or something. So then I sort of stopped on it for a while, and then I kind of rebooted and wrote an NSF grant that was describing this idea of the agent-based modeling, a little bit of how to model adaptation. So it was really like kind of more basic, fundamental, not really the clinical problem. And then now, I think it's been, since I wrote the first grant, I think the first grant I wrote was in 2007. And so now I have 2 grants from the NIH on it. So it took like a long period of time. But if you kind of keep on with it, do plan B, then you ultimately get there. So anyways, modeling is an adventure. It's pretty noisy. Who knows? Each project's going to have different. And you never really know what the impact. It's going to take a while to know what you're doing. But I guess hopefully there's like an upward trend of impact. So just to, I wanted to thank a bunch of students and collaborators. I don't think I even got everybody's name here, but Adrian is here in the room, and other students who really contributed to this, Kelly Virgilio, Zhao Hu, Kyle Martin, Bahar Sharafi, Katie Knaus, Nick, and Nick, who was here for a while. Now he's moving off to bigger and better things. And our collaborators, our funding sources in the lab. Thanks. Oh, am I looking for a postdoc if anybody wants a job?